All right, it's time to start the session. Hello, everyone. Welcome to our webinar, Decoding Bacterial Host MGE-associations with Scalable Single-Cell Genomics. I'm Žana Zdanienė, Director of Development at Atrandi Biosciences, and I'm pleased to be co-hosting this session with Mária Džunková. Welcome, Mária. Mária leads the Mária leads the microbial single-cell genomics group at the Institute for Integrative Systems Biology at University of Valencia. In her work, she uses single-cell as well as metagenomic approaches to study microbial ecology, uncover new microbial lineages, and analyze bacteria-phage interactions. So we will start this session from a brief introduction to Atrandi Biosciences single microbe DNA barcoding kit, including the underlying technology, the workflow, and the application to environmental samples. I will be presenting this part, and then I will hand over the virtual stage to Mária, and she will discuss how her lab is using high-throughput single-cell sequencing to study active phage infections and host associations in various ecosystems. So let's start from the workflow overview. As you may know, shotgun metagenomics has revolutionized microbial ecology by enabling the analysis of unculturable microorganisms. However, this method is limited when it comes to highly diverse samples. Genomic assembly is quite complicated when genome abundances vary, taxonomic resolution is limited, and the ability to link function or mobile genetic elements to their hosts is very complicated. To address these limitations, single-cell sequencing offers the analysis of DNA extracted from individual cells, enabling the precise and direct host association with mobile genetic elements and more accurate and precise genome recovery. There are two main approaches currently used in the field. So first one is phage-based sorting of individual cells into plate wells. This approach is quite convenient in terms of the compatible lysis options and assay designs. However, it hardly scales. On the other hand, droplet microfluidics enable easy scaling, but the choice of lysis options and molecular biology assays is quite limited due to droplet stability issues. Here at Atrandi Biosciences, we have developed a so-called MSPC, a workflow based on semi-permeable capsule technology, or SPCs for short. This approach offers the scalability of droplet microfluidics, but retains the flexibility and robustness offered by plate-based approaches. So I will describe the underlying technology behind MSPC. This is semi-permeable capsules, or SPCs. SPCs are miniature compartments that we use to isolate individual cells. These compartments are emulsion templated, meaning that at the very first step of SPC generation, we are creating microfluidic droplets using two aqueous phases. Those aqueous phases contain special polymer solutions, and those polymers are immiscible. Once the droplet is formed, polymer solutions start to phase separate. To stabilize this so-called aqueous two-phase system, we expose droplets to light, and at this point, the shell polymer that is located on the outer surface of a droplet solidifies and becomes a very stable porous hydrogel. Once capsule is formed at this stage, it exhibits a lot of properties that are advantageous for microbial workflows. So first of all, this hydrogel shell is very chemically stable, so you can use various harsh chemicals to lyse the cells. For example, alkaline lysis buffers, lytic enzyme cocktails, temperature treatment. And another property is size-selective permeability of this hydrogel. This means that once the cell is lysed, large molecules, such as genomic DNA, will be effectively trapped inside the capsule, while smaller molecules like enzymes, short oligonucleotides, and buffer components will be able to freely diffuse in and out. So practically, this means that capsules enable multi-step sample processing with very easy reagent exchange between individual steps. So based on this technology, we have developed a single microbe sequencing workflow, which starts from processing of biological sample to obtain a cell suspension. At this point, we need to remove any debris that might clog the microfluidic chip. Then the cell suspension is being encapsulated into semi-permeable capsules, and we have a device called flux designed for this purpose. Once cells are inside SPCs, we then transfer them into a regular test tube, and all downstream steps are performed just by manual pipetting in regular test tubes. We then proceed to whole genome amplification, DNA fragmentation, and DNA end prep. To label individual cells with the unique barcodes, we're using combinatorial barcoding approach that consists of 4 sequential rounds of barcode ligation. Before each round, we mix all SPCs and then randomly aliquot them into 24 wells of a barcoding plate. And each well contains a specific unique barcode sequence. After 4 iterative rounds of ligation, each individual SPC ends up with a unique combination of those 4 barcodes that eventually will enable us to trace sequencing reads back to the cell of origin. So first of all, to test the workflow, we performed a quite classical species mixing experiment using a very simple mixture of Escherichia coli and Bacillus subtilis cells. And at this point, we observed that indeed DNA retention in SPCs is very good. We observed very minimal barcode cross-contamination level between SPCs. So meaning that we can actually achieve single cell resolution. Upon reference-based analysis, we were able to recover more than 90% of genomic information from individual cells at coverage depths of 10x or higher. And once we analyzed the same data set using reference-free approach, so meaning de novo assembly, we were able to recover near complete genomes from individual cells at coverage depth of 20x or higher. We then challenged the workflow with real environmental samples. Those were river water sample and soil sample collected in Vilnius in Lithuania. Both samples were processed using 2 approaches, so single cell sequencing and traditional shotgun metagenomics. We prepared a few metagenomic libraries that differed in terms of lysis conditions. In the slide here, AL stands for alkaline lysis and BB stands for bead beating. And I must note that for single cell sequencing, we used only a very simple alkaline lysis buffer to establish the baseline performance of the kit. And of course, we know that alkaline lysis buffer is not optimal for gram-positive bacteria and other species that are hard to lyse. But good news is that the workflow is flexible in terms of optimization options, and it's very easy to adjust the lysis conditions by adding other chemical agents, lytic enzyme cocktails, or temperature treatment as appropriate to your sample. Taxonomic analysis of our samples revealed that in river water sample, we had roughly around half of the contexts that were unclassified, which is normal for environmental samples. And among those that were classified, we observed that single cell data provided a broader taxonomic diversity, as evidenced here in Venn diagrams. Venn diagrams show the overlap of taxonomic categories at different levels, and here single cell data is represented by a purple circle. We also checked which organisms were detected, and the results were ecologically plausible, so meaning that the data basically made sense for the source of microbial community. In soil sample, the pattern was very similar. Again, a big chunk of contexts were not classified, but among classifiable contexts, we again obtained a broader taxonomic diversity in single cell samples. And again, among those organisms that we were able to detect from the biological and ecological perspective, the results made perfect sense. We also searched for interesting functional genes like antimicrobial resistance genes, virulence genes, and plasmid sequences in our data. And this slide shows an example result from the river water sample where we actually detected a bunch of virulence genes. We also detected a few plasmid sequences. And this graph here on the slide shows a single cell dataset and illustrates how easily we can trace back those functional genes to their host. This is something that is just impossible with bulk approaches. So in metagenomic data, this linkage is lost. Even more striking example of this host MGE association was demonstrated in a separate study where we sequenced single amplified genomes from human oral microbiome sample. In this case, first of all, we found a lot of viral sequences, and in single cell dataset, we were able to trace them back to their hosts. And interestingly, a subset of single amplified genomes contained both antimicrobial resistance and virulence genes, as well as plasmid sequences. And some of those genes were on plasmid sequences. So meaning that these findings may serve as indicators of potential clinically relevant dual threat pathogens. So taken together, these pilot studies highlight how high throughput single cell sequencing enables the functional and taxonomic resolution. This workflow is now commercially available through Atrandi Biosciences. We do have a flux system and SPC generation kit for easy cell encapsulation into SPC. We also have single microbe DNA barcoding kit that contains reagents for cell lysis, genome amplification, barcoding, and Illumina-compatible library preparation. The kit is designed to process up to 10,000 individual cells in a single experiment at a very reasonable price per cell. For more information, please visit our website at www.atrandi.com, or if you have immediate questions, please feel free to post them in the chat. We will address those questions at the end of the session. So thank you for your attention to this introductory part. I now hand over the stage to Mária. Mária, you're welcome to proceed with your presentation.
Hello, thank you very much. I would like to thank Atrandi for inviting me to give this presentation, and I thank everybody for coming. So let's go. I will present you my results that we obtained in our group during the last few months using the Atrandi platform. So I will just tell you briefly what are our objectives in my group, in the microbial single-cell genomics group in the Institute of Integrative Systems Biology in Valencia in Spain. So basically, we use very often flow cytometry to study functions of uncultured bacteria in their natural ecosystems. So we obviously, you know that in the natural ecosystems, there are a lot of bacteria that are so difficult to study that it's actually not very worth trying to isolate them and study them as culture isolates. But since there are a lot of techniques how you can assess what they actually are capable of doing and capture them in that moment when they are producing some special metabolites or are, for example, covered by immunoglobulins or different things. So actually you can use fluorescent probes and you can detect fluorescently the function of your interest. And by using the flow cytometer, so you just collect the bacteria with these fluorescent tags. In the past, we were using this in a way that we were sorting our fluorescent cells into 96 or 384 well plates. So the logical option is that since we have them sorted by the flow cytometer in individual wells, so the logical option is that we will do single cell genomics on these individual cells. So in the past, I was, Okay, so during my postdocs, where I learned these techniques, so I was working in a quite rich laboratories that I have a lot of robots that were helping to deal with these single cell techniques. This is very important because when you distribute the liquid solutions, lysis buffers, whole genome amplification reagents, you have to be very careful because they can be some contamination by the air from your own DNA and so on. So actually what you think that is single cell can be actually highly contaminated well. And so this is very dangerous. So I started during my first months in my current lab, just using a multi-channel pipette. It was it was very difficult times, but I was so lucky that just right during that time, so I was contacted by Atrandi, and actually my life changed a lot. But I just would like to illustrate what was our workflow before I started to use Atrandi. So we were monitoring the whole genome amplification on these 96-well plates. This was very important because the typical reagents are not performing properly in all the cells in the natural environment, because some of them are not lysed properly, or maybe the whole genome amplification is not working properly. So if you are thinking of like working individually on a 96-well plate format, you have to think that every single cell will be sequenced individually. So if you have some experience with Illumina sequencing, for example, you can do some kind of multiplexing, for example, combining 96 samples in one plate. But of course, you don't want to risk, because each individual sample costs quite a lot of money. So sometimes we were doing some double checking before sending them for sequencing. We were even doing like some small checks by amplification of the 16S gene to make sure that what we see as amplified curve is actually a real bacterial genome, because it could be just like some piece of DNA of bacteria which is amplifying a lot. So that would result in a lot of expenses, a lot of money which would be wasted for sequencing non-existent genomes. So after that, I purchased Atrandi Flux system, which really changed our life in the lab. So right now, of course, we have to think very well what kind of environment we want to study, because this will generate a lot of data. So this is a huge change in comparison with what we had before. So now we can process 10,000 cells in a single run, and I have calculated that this amount of cells we will be able to process in 8 months, because as you can imagine, one cell in one well of the plate, this means only 100 cells per day, and all the associated checks and everything. So at the end, it will be quite a lot of months actually of work, and the cost of the cells would be it's also incomparable. Right now with Atrandi is much cheaper. So as you heard from Žana, so the biggest advantage of this system is that it has semi-permeable capsule. So actually you work in a single tube with 10,000 cells at once, in the same way as you would work with your normal metagenomic sample. Yeah, like you can do extractions and any other kind of workflows, because everything can be done there, since there are pores that allows you to exchange reagents. You can do PCRs, and it's super resistant. So I'm really amazed. In our lab, so we have set up 2 different ways or manners how to deal with our single cells, in which we implement Atrandi microfluidics. In the first one, so this is the classical workflow that Žana presented. So this is that you have your environmental sample. There are 2 ways. So you can just like take the sample and calculate more or less. There should be like 62,000 cells to begin with, and then with all these washes and so on. So they guarantee that you will have at least 10,000 single cells. So you can calculate this number by just cell dilutions, or you can go to flow cytometer and actually implement some specific labeling. If you have like some fluorescent antibodies or any kind of fluorescent label, so you can just select the bacterial population of your choice, and you collect them in a 1.5 milliliter tube and microliter tube, 1.5 milliliter tube, and you then just proceed with the Atrandi protocol. And here, so in order to get every single cell tagged by individual barcode, so you have to do split and pull barcoding, and at the end, you end up with more or less 10,000 barcoded cells. And then I will tell you about our program CleanBar that we developed for optimal demultiplexing of these barcodes. In the second workflow that we have on the right side, so there the steps are a little bit changed. So first we encapsulate our microbial population, then we use the whole genome amplification reagents to generate a huge amount of DNA template, and then we can use some fluorescent labeling. In this case, this is PCR labeling, so labeling with fluorescent primers, and then we do sorting. So this second option is more suitable for the workflows where you don't use any fluorescent label targeting the cell surface or some cell activity, but you can use your favorite PCR primers, and the only thing you have to do is just to add some fluorescent tag. And these fluorescent primers can be used like in a normal PCR, but here you apply this PCR to the tube in which you have your capsules, and you just do the PCR, and after this PCR you obtain a lot of fluorescent amplicons. So these amplicons generate a lot of fluorescence, and then you can go to the flow cytometer and collect the cells that are fluorescent. So obviously you don't want to collect 10,000 cells because this is crazy, but you could not do sequencing of 10,000 individual wells. But if you are interested in doing just like some small scale sampling, so you can collect like, I don't know, one plate and sequence them separately, as I mentioned, by multiplexing on Illumina platform and so on. So depending on your objectives, on your throughput, and on which kind of fluorescent labeling you prefer, so you can choose one or the other workflow. So first I will talk about this encapsulation, the LC approach, which is label, sort, encapsulate, and amplify. So this is this high throughput option where you have the whole community. And since I'm very interested in the connections between the phages and the bacteria, how do they interact? So I will show you the results of our pilot experiment. And in the second part of my talk, I will explain you one example that how we are trying to track mobile genetic elements in the bacteria, in the symbiotic bacteria of nudibranchs. So first of all, I will explain you in more details what actually is this split and pull barcoding. So imagine that you have all your encapsulated single cells in one tube. You just aliquot the equal volumes in the first portion of the plate. So this is 24 wells, first 24 wells. You do the ligations because in these wells there are the specific barcodes, and then you collect everything. So you really pipe it very well, everything, and pull it, mix it, and then you distribute it in the second portion of the plate, and then repeatedly do the same thing. So actually here we have like some zoom into what is happening in the individual well. So in the first round of ligation, for example, this pink well, so obviously all the single cells, they have this pink label, but then they are mixed. So we can see later that, in the second row, the last capsule, which is on the right side, so it has already the pink label and the red label, and so on. So at the end, it's impossible that one capsule, that there will be two capsules that have the same label. So there is much more combinations created by this split and pull barcoding approach than you have your cells in this assay. So you start like with 62,000 more or less, and you end up more or less with 10,000, but there are like more than 300,000 of combinations. So it is ensured that this really provides single cell resolution. And these barcodes, so they are connected by ligations. They have some linkers. So there are 4 base pairs linkers that this is what actually facilitates the ligation. And the fragment that you obtain, so some of them, in the ideal case, both of the ends will have the barcodes, but since the sequencing is not perfect, so you just expect that all your fragments will have the barcode at least on one end of the sequence. So in our pilot experiment, sorry. Yeah, okay, let's go slowly. Okay, so in order to do the demultiplexing, we have developed clean bar tool. So this is a very simple program that is actually pretty flexible and produce a lot of interesting data that helps you to go through all the errors that you might commit during the ligation step. So it has been originally developed to Atrandi barcodes, but if I know if any other barcoding, split example barcoding approach exists, or maybe Atrandi will change it in next month. So this is just very flexibly can be adjusted to any kind of barcoding composition of the barcodes and settings and everything. So in this. In the case of Atrandi right now, so we have the total length of 45 base pairs, because actually there are barcodes of 8 nucleotides, and the linkers between them are of 4 nucleotides. So our questions that we just wanted to ask in the beginning in our pilot run was that whether all the sequences will have the 4 consecutive barcodes, and if not, so can we do something to save the sequences that do not have all the barcodes? Then is the linker length always the same? Because, you know, if you know the sequence, the linker sequence and the whole sequence, so actually you could just like do some quick blast and so on. But what if this ligation is not totally perfect? So happens something like that or not? This was our question. And actually, can we track that all our barcodes have the same efficiency? This would be very good for like a feedback to really benchmark like whether we have some problems in our pipetting or our laboratory issues. So now this program works like this, that you just have your original FASTQ file, which is produced by your sequencing platform, and then you have to provide a barcode list, which is a simple text file. And you will get output files. There is some search, the output of the search that is visualized on your screen, and you get some summaries, tables and statistics on like what is the proportion of the barcodes and the length of the linkers and so on, to make sure that everything was perfect correctly. And then obviously the objective is to get the demultiplex files. So every single file, so you will obtain the single cell demultiplex files, which will obtain the names according to the barcode labels. So for example, this A10, H7, A4, B1, this is the label from barcode D, C, B, and A. And also we can capture and gather the cells that accidentally did not have the full barcode. So they can be useful for some application as well. And also we gather the sequences that do not have any barcodes. So in our experiment, we used Klebsiella phage interactions. So we had, because this was our objective, really wanted to test whether this assay is able to distinguish between 2 different species and strains. So we had 2 different Klebsiella species and 2 different strains of each of these species. And we had phages of different morphologies. So we exposed them. We expose the strains to the phages and do the control infection experiment. And we also added some negative control phage. So this is also very important, because just imagine in the natural environment, you can have some accidental phages which are attached, but actually they do not enter the cell. And maybe the reagents that we use for fixations can also interact and maybe create some fake, like some interactions that are not real. So we wanted to make sure that our workflow is correct. And we use the facts to count exactly 15,625 cells, which multiplied by 4 is equivalent to 62,500. And so this is, as the protocol says, you need this amount of cells to end up with 10,000 barcoded genomes. I have to say that we were very happy to be one of the first researchers to test the new optimized version for the PAC biosequencing. So here the whole genome amplification reagents of Atrandi is optimized, adjusted to produce longer reads. So we finally obtained 0.1 micrograms of DNA with a peak fragment size between 5 and 5.8 thousand nucleotides. And our sequencing run on PacBio generated 2.3 million reads. So the output was pretty good, obviously not enough for covering all the Klebsiella genomes, but you will see that we got pretty good results. First, I want to show you how the clean bar program works. So this is the screen output, where you can see how it aligned, that it found the barcodes nicely separated by 4 base pairs linkers. It prints here the barcode label, and here it prints the barcode position as well. And one important thing is that we process these reads coming from the PacBio platform. The adapters, the sequencing adapters of PacBio should be eliminated by the program, but actually it did not work properly. So this is the good thing about clean bar, that actually it works independently on the beginning of the sequence. So it scans it from the beginning, but also is very careful because some of the barcodes can just accidentally appear in the sequence because they could be naturally present there. But it's impossible that in nature, these 4 barcodes, these 4 sequences of 8 nucleotides appear in this distance and in this order. So this is obvious that this is the real barcode. And in this case, it was found in the direct sequence and also in the reverse complement sequence. So the whole PacBio run was barcoded perfectly from the both ends. So here we have the results of our pilot run. So we have found 11% of the sequences that had the beautiful 4 barcodes. We also had some of them that had only 3 barcodes. I will show you later. I think this was our pipetting error. I think this is not very usual, but this is why we use this program, and this is the result of our pilot run. We also had some sequences with no barcodes, but this was actually our original decision or our experimental design, because we did not do any enrichment to enrich for the sequences with barcodes. So actually, maybe you did not even realize that, but when you do like when you prepare some Illumina libraries, so you always enrich for these for the sequencing with the sequencing adapters. So actually there are a lot of sequences that you lose, and actually this is totally natural that this happens. And I think that in fact this number in our results is not actually high. I think this is pretty good result. Sometimes we also find some skipped barcodes, so this is also our fault, like that we did not do the ligation properly. Maybe some barcodes were just lost. But we can still use this read for some other applications. And the interesting thing is that I was always saying 10,000 cells, you will get 10,000 cells, but we were able to get 50,000 cells. And this is actually, this reflects our ability to pipette very efficiently. So maybe we were losing a lot of time by recovering all the materials from all the wells. So we recovered 15,000 single cells. So this is pretty good, and so this is more than expected. And but on the other hand, I will show you on the other slide that this portion of the plate with the barcodes starting from E1 to H3. So these barcodes were not ligated properly. So probably here there was some error during our pilot run. But anyway, so this is the result. This is how it went. And I guess that next time we will perform better. Here we also, as mentioned, we could reveal that not all the sequencing adapters were trimmed properly. So the barcodes did not start at the position 1 of the read, but actually this is not a problem for our program. And most, actually like 99 point something of the linkers were of length 4 base pairs. So this is as expected. There are some very little errors, or some maybe also due to sequencing, that some were just of 3 base pairs or 5 base pairs, but really not a problem. So this was also very satisfactory. And here are the results of our Klebsiella phage experiment. So we had 4 strains, and here you can see in this bar plot in how many cells we obtained bacteria only. and how many of them were bacteria and phage, and in which of them were phage only. So this is very interesting because actually I mentioned you that we were using fluorescent activated cell sorter to collect bacterial cells. So we were sure that everything what we were collecting was of bacterial size. So it doesn't make sense that there are phage cells only. So I will talk about that in my next slide, just to draw your attention to this phenomenon. So since we obtain more than expected number of the FASTQ files, you might say, okay, so many of them are multiplets. So it means that accidentally 2 cells are inside of one capsule and error like this, but actually everything was as expected. Our multiplets were even lower than expected. And since our coverage was not that high, so we had more or less like an average, like ranging from sometimes in some cells were only 1 read, but in some were 533. This was our maximum. Median was 3 reads per capsule. This sounds pretty low, maybe you would say, but not. We obtained quite a lot of single cells that had pretty enough coverage to study the phage and bacteria interactions. And only 0.30% of the reads align with more than 1 bacterial genome. This is also very good because this means that we can distinguish between very similar strains, and this 0.13% can actually reflect the natural similarity between these strains. So this is also very, very good result, and gives us the certainty that we can really obtain the single cell level resolution on the strain level. So why we had these phage only capsules? I think that this is because these phages have very high burst size. So it means that they produce a lot of copies when they infect the bacteria. So when we have a low coverage, so it means that we preferentially recover the genome of the phage because it's present multiple times in the capsule. And so when we have low coverage, so it means that we just capture the reads coming from the virus and not from the bacteria. But the very good thing is that even if we had pretty low coverage in our sequencing experiment, we were able to detect the phage host interaction in the capsules that had as few as 5 reads. And the proportion of the phages in these capsules, the capsules that had phage and host pairs were almost 40%, and they exceeded even the capsules that were bacteria only. So it means that we really captured this community in the moment that they were exposed to the phages and they were infected, but it also reflects the heterogeneity of this infection. So even if this was a clonal population coming from one culture, so within these populations, there are bacteria that are reacting differently to the phages. So this has been observed in different ways, like some qPCR quantifications and some culture experiments. But now, so we can see it in real DNA sequencing data, and it matched our culture experiment. So overall, I'm pretty satisfied with this host phage interaction detection. The very good thing about Atrandi chemistry is that it produced pretty uniform coverage. This is not usual for the whole genome amplification kits. So I was very happy to see these results. Actually, it was possible to observe the coverage differences between the genome and the copies of the phages on the single cell. And so we were able to estimate more or less some kind of burst size, or let's say the number of the copies that were formed and captured within the single cells. So this is something that you could do when you have like bulk resolution. So this is the typical experiments that are performed with some metagenomes. But now, so we have single cell resolution. So actually, we can see this heterogeneity within one population of bacteria. So the summary of this experiment is that we had not many reads, which would yield, in total, they would yield like 1.6 thousand coverage of Klebsiella genome. But when you think in single cell resolution, so actually this is not enough to recover the whole genomes of each individual cell, but this was not necessary because we had the reference genomes. But we just demonstrated that even if we don't have enough coverage, so the cells that contain at least 5 reads are sufficient to track these phage and bacteria relationships. We were able to find the single cells that had pretty high content of phages inside. And we were able to distinguish the strains level in these capsules. And the important thing that we did not detect any negative control phage. So this gives us green light to go to do the sampling in natural communities. Like obviously designing your experiment with the natural community, so you would have to think about the read depth you want to obtain. If you don't have the good reference genomes to rely on mapping, so maybe you can fabricate or obtain your own, because these reads obtained by the Atrandi platform, when you trim the barcodes, you can actually use them as normal metagenomes. So you can actually use your these sequences to generate your own metagenome assembled genomes and then map your single cell resolution reads to these bags and maybe study the single cell on single cell, the strains, and so on. And then I will just briefly comment about this encapsulate, amplify, label, and sort approach. So it means that you first encapsulate, do the whole genome amplification, and then use this DNA as template for your fluorescent capsule PCR, and then do the sorting. So we focus on, in this workflow, on nudibranchs. There are these beautiful marine soft-bodied animals that are of our interest because we have recently discovered that there are some very interesting symbiotic bacteria. In the past, so it was believed that these nudibranchs, they produce some molecules to defend themselves against their predators. There were also some evidence that they can recycle these molecules from their prey. In this case, it's usually a sponge, and produce the molecules, but we have found out there are some bacteria that can do it. So actually, we have found, we have discovered several new bacterial, symbiotic bacteria that belong to new orders. And now we want to study what are their interactions with their hosts and how do they contribute. So we have mined the biosynthetic gene clusters in these genomes. And to our surprise, we have found one which was present on the plasmid. So this is not unusual. The biosynthetic gene cluster can appear on the plasmids, but what was interesting here that we found the same cluster in some strains in the genome and sometimes on plasmid. So, and since we know that these nudibranchs are very efficient in recycling the material from their prey, in this case of sponges, so we wanted to know whether this plasmid is transferred through the bacteria, is this the same bacterial host, and so on. So I think this is the good experimental question to ask and to address by this Atrandi platform. So just imagine, so these are some images that we have from our encapsulation. So we have amplified the DNA. So we actually go from femtograms of DNA all the way to micrograms. And so we amplify the DNA, which is in there, use it as the template for the PCR. And if there are some plasmids or phages, here visualizes these green circles. So we will use the PCR fluorescent primers to track them and actually can find that this green plasmid is not only present in green bacteria as expected, but also in some orange one, different genera, or, you know, it could be very interesting. So the ideal thing would be to purchase the special sorter from Atrandi, but we don't have budget for that. So we were trying to adjust one special flow cytometer that we had at our university, because these capsules, if you use the Flux platform, so there are too large for the typical fluorescent activated cell sorters. So you have to use some special ones, or you have to generate the capsules, which are smaller. So this is another instrument. Maybe Žana can explain this later. So yeah, the sorting is a little bit challenging, but we finally figured out how to do it. And here in this plot, you can see that how we found the capsules, and then we labeled fluorescently by the PCRs actually by the PCR primers, targeting our biosynthetic gene cluster. So we obtained the fluorescence and collected these cells. So this is my group at the University of Valencia. And so obviously I have to thank our funding agencies, and thank you for attention. I'm ready for your questions.
Thank you, Maria. It was a great talk. Let's see. So we do have 15 minutes for questions, and I'm just opening the chat to see if we have any. Let's see. Okay, so maybe I will start from the questions to you, Maria, regarding your recent talk. So one of the questions is, how efficient is SPC sorting with phages?
In phages? Fluorescent activity cell sorter?
Yes.
But what do you mean, like how efficient?
I cannot comment to the whole question, but perhaps if you have some experience with sorting those large capsules, you can comment on that, and I can explain how we can make it better.
Yeah. So I have in my lab only this flux instrument for microfluidics. So it generates the capsules that are 70 microns in diameter. So the problem when you have like many laboratories or core facilities for phages, like the typical phages is, for example, phages area, so actually these instruments, even if you use the nozzle, the widest nozzle possible, so it generates quite large capsules, but you have to consider the cell to capsule ratio, and we had some problems because the beads that are used to calculate the drop delay, so they fall in different speed. So it's because these droplets are of maybe different weight or something, but it just does not fit. So there are some experiments that you have to collect your supposedly fluorescent particles, but actually we did not count we had to count or collect like 50 droplets with our capsules, but we only collected one, and it was because the drop delay was incorrect. So I think that one of the possible solutions would be just to purchase another instrument from Atrandi, which is where we can generate smaller capsules, or maybe this would be my dream to to purchase the sorter directly from Atrandi for these capsules.
Yeah, I can comment more. So you're absolutely correct that the SPCs that are currently generated on Flux are too big for most of the phages devices currently available commercially. So one option is to, as you mentioned, to generate smaller SPCs, but for this you would need another device called Onyx. We do have it available. It's just an alternative system that is more flexible in terms of SPC size, throughput, and other microfluidic parameters. And we do have an SPC generation kit that is capable to generate capsules of 30 microns in size. So this size is actually perfectly suitable for most of the phages machines with regular 100 micron or 70 micron nozzles. Another device that you're talking about that we do have, so we have a system for droplet sorting, and you can use it for SPCs and for larger SPCs as well. And I also can comment that we are developing a new system which will be called phage, that will be able to sort even larger capsules, larger particles, up to hundreds of microns in size. So this is something that's coming up. We do recognize that it's a problem, and in many workflows, sorting of capsules will be a very advantageous step. So we will keep everyone posted on the progress of the development of the system. Okay, thanks, Maria. I do see another question. So how does the throughput of the Flux compare to that of the Onyx? If I encapsulate my bacteria using Onyx instead, can I expect the same throughput of 10,000 to 15,000 cells after the barcoding? So I can address this question in terms of the throughput after barcoding. There would be no difference because the input, the SPC input into the barcoding workflow, we measure it in terms of the volume of SPCs that you need to collect before going to the ligation step. And on Flux, you can generate it either on Flux or on Onyx. On Onyx, in principle, you can tune the throughput, you can collect more capsules, but then you need to be careful and to calculate if you want, in order not to overload the plate, because in order for combinatorial barcoding to work, you need to keep the number of occupied SPCs much smaller than the number of all theoretical possible combinations of barcodes. So I would say that, well, in practice, SPCs generated with both instruments in this regard will be equivalent. Yeah. Okay, we do have another question, perhaps regarding to my part of the presentation. How do I choose correct sequencing depth for soil samples? This is an interesting question, and I don't think I will have a very concrete answer. It depends perhaps on your application. Do you want to assemble genomes de novo, or do you need to just perform some taxonomic assignments? We typically recommend to aim for around, at minimum, 100,000 reads per cell if you don't need the full genome recovery, and around 2 million reads per cell if you aim for de novo assembly. So that would be the ballpark. Another comment regarding the sequencing that I perhaps failed to mention in my part of the presentation is that upon barcoding, you are not obliged to sequence all 10,000 cells. So you can sequence just a subset of your capsules, for example, 100 cells, 200 cells at a greater depth. And then if you see that there's some interesting organisms, or you would, get your biological answer even from a shallower sequencing, you can go ahead and sequence the rest of the capsules. So this, in this regard, the workflow is quite flexible. We have another question related to sample prep. Is there a way to deplete certain cells before sequencing? For example, if I have a lot of host cells in my sample, is there a way to remove them to only get bacteria? So that's a good question. We don't have a direct experience with host depletion, but I would imagine that it would be a part of the sample prep, because there are kits for differential lysis when you can, for example, lyse eukaryotic cells before moving to encapsulation of bacterial cells. But I don't know, maybe Maria, you have more knowledge and experience on this part.
Yeah, so definitely. So there are two ways how this can be done depending on who is the host. So I don't know, like in the first instance, so maybe you can do something very simple, which is just depending, like if your host cells are large enough and your bacterial cells are the typical bacterial size. So you can just do, some filtering by 5 micron filters. So this will at least remove some portion of the host. And then you can still, with this prep of cells, you can go to flow cytometer and maybe there refine your sorting, because the kit of Atrandi recommends to start with 62,500 cells, 62,500, something like that. And so then, so you have to have this number, and I think that just one session at phage and trying to enrich by size sorting for your bacterial cells, that will be also an option how to proceed before you start with the barcoding. And then at the end, so when you do barcoding and you have everything lysed and so on, as Žana said, so you can also use some special lysis buffer, which is ignoring your host cells and preferentially lysing your bacterial cells. But if this is not 100% efficient, so you can still use some other kit that is targeting your host DNA. So there are some probes that, for example, there are some streptavidin beads that, depending, yeah, so you can either prepare them yourself. So you can prepare some probes that will hybridize with your DNA of the host, and then you can kind of collect everything what will be discarded, and you will just sequence the reads which are barcoded and belong to your bacteria. So I think there are several steps that you can do this. Or maybe, yeah, also, actually, you could, sorry. So I have an idea that you could actually do yeah, like when you have your barcoded DNA, and you have a mix of host and bacteria in your sample, so you can do like this fluorescent maybe PCR to collect only the bacteria, or to remove the host maybe, and after that you do the you do the step when you dissolve the capsules, maybe, yeah, something like that. So there are different levels.
Yep, thanks a lot. So there are strategies. Yeah, and I think at least some of them are definitely practically feasible. We do have other questions. So one related to PCR primers, perhaps the ones that you use for enrichment of your target sequence for sorting. How do you stain the primers for PCR sorting? So is the fluorescent label on located on the 5' and on both forward and reverse primers, or not? What is the primer design?
Yes, it is on the board, and this is something that you can. So I use IDT website to design my primers and to place my order. And there is a way how you can just choose some fluorescent label. And obviously, these primers are much expensive. Like one costs like 250 euros, more or less. But maybe you can find some cheaper options. And yeah, so this is how we proceed.
Yeah. Okay. Another question about smaller SPCs. Is the 30-micron SPC kit available for flux, or when do you expect to release that? So perhaps it's a question for our side. For now, no. We only have a single SPC generation kit that allows you to generate around 75, 80-micron SPCs on flux, but we do recognize that there is a pressing need for increasing the throughput of SPC generation for certain workflows. So the development of a broader variety of SPC generation kits for flux is definitely on our roadmap. Unfortunately, I cannot give you the exact date, but we will definitely keep you posted. And yeah, I think in upcoming years, we will come up with more kits compatible with flux. Okay. Did you characterize how DNA is retained in SPCs as a function of length? What is minimal DNA length retained in SPCs? I can answer that as well. In our internal experiments, we typically tend to measure SPC permeability in terms of the DNA, double-stranded DNA fragment size that is either retained or can diffuse. And we see that full retention is obtainable for DNA fragments that are 500 base pairs in length or longer. There is a kind of a gray zone. So from 200 to 500 base pairs fragments, they are somewhat retained, but they start to diffuse if you would keep them in capsules for longer periods of time. So in principle, you can go down to, for example, 300 base pair amplicons, but with a slight risk of cross-contamination. But what is lower than 150 base pair will diffuse in and out freely.
Yeah, I can confirm that. Yes.
Yeah. We do have another question for Maria, for you specifically. Could you share what future directions you see for this technology in your lab, and what big biological questions you hope it will ultimately help you answer? The future looking.
Yeah, so I'm not very sure I want to talk about that because yes, like I would like to look at some unusual mobile, like the DNA traveling in some unusual ways. So not necessarily plasmids or phages. So that will be to assess the some alternative ways how the DNA can travel between bacteria. And I'm also looking forward to see how which different kits or different approaches, like chemical reactions, I can use the Atrandi for, especially the microfluidics. And then because since they claim that it's very flexible, so we have different laboratory procedures that could be easily adapted to these microfluidic encapsulated cells. So we are not limited to genomics. So we can do different things. And actually, we did not even talk about culturomics and that how useful it is for culturing bacteria. So this is also quite interesting that you can you can move from the population level in your biological questions to the single cell level, and you are not limited to genomics.
Okay. Thank you for the answer. Our time is running out, but we still have one question to answer. Can cells be fixed prior to sorting to prevent ongoing processes such as viral infection before encapsulation, or would fixation compromise subsequent sequencing steps? I don't know, Maria, do you have any experience with
Yeah, for sure. So I before I did this pilot experiment, so I already knew that formaldehyde fixation is bad for whole genome amplification. So, and I knew that a better alternative is metacar. So this is some mixture of methanol and so on. Actually, methanol is used anyway in the Atrandi protocol in the beginning. So we fixed our bacterial cultures exposed to the phages with metacar. And so this was actually our objective in this experiment. We wanted to make sure that when we use this fixation, because usually the fixatives, what they do is that they cause some clumps and, that you can maybe get some, doubles of, actually they are not single cell, but there are two cells or several cells in one droplet. So actually, it did not affect the performance. So I would definitely recommend metacar.
Okay, that's very useful piece of information. Yeah, thank you, Maria. Okay, so that concludes our webinar. Thanks a lot, Maria, for joining. It was great to having you here as a co-host. Thanks everyone for joining. I hope to see you in our future webinars and events. But for now, thanks a lot. Have a great rest of the day. Goodbye.